So we finished off glycolysis and said there was an in-between step before the next stage. And so we're going to talk about the citric acid cycle next. The citric acid cycle is aptly named because it has citric acid as a molecule in it. It's also called the Krebs cycle, after the scientist who developed an understanding of it. If you are to watch the extra resource videos that are available to you on Moodle, there is the Crash Course series, does some good history on the scientists that developed a lot of this understanding and give you a bit more context behind who Krebs was. And that can sometimes help some of these names stick for you. It really doesn't matter whether you call it the citric acid cycle or the Krebs cycle. Both are absolutely fine. But it is important that you kind of connect with the meaning of these. They're named for a reason. And so the citric acid cycle, we're going to start with acetyl-CoA, which is why that's what the notes say, and that's not where glycolysis left. So we did have that in-between step that was neither the Krebs cycle nor the glycolysis nor the Krebs cycle. And the whole citric acid cycle is really a series of carbon adding and removing steps. And so it is a cycle. It literally cycles through and keeps going round and round. And as the carbon gets added and taken away, there's a few key things that happen. Overall, carbon dioxide is going to be produced like we saw in that in-between step. When the carbon combined with oxygen, it created carbon dioxide, and carbon dioxide is one of the products of the overall cellular respiration reaction. It requires oxygen for that reason, right? We are now in the mitochondria. So we've moved from the cytoplasm for the glycolysis stage into the mitochondria. So the mighty mitochondria is where we're talking about now. That you would have written in your chapter 4 stuff that the mitochondria is the site of energy production, except for the glycolysis. That's technically in the cytoplasm. So the other things that happen in this citric acid cycle is that it's going to produce some ATP, and it's also going to produce some more NADH, which we met in glycolysis, and a new molecule, this FADH2. So both of these, I always add in the notes, they are electron carriers or energy intermediates. As I had stated in the glycolysis discussion, these molecules don't show up in the chemical reaction because they're used in the process. They're intermediate to the process. They're going to be used in the third stage. They're going to be used in oxidative phosphorylation, which is coming at us here. And so I often do this. I'm going to switch to a big scary picture of the citric acid cycle, and then we're going to bring it back to a simplified version, really focusing on the key things. So this is the big scary version of it, and you can see that we have the acetyl-CoA going into it. And I wouldn't concern myself with the other names of these molecules, but it combines with a 4-carbon molecule, making a 6-carbon molecule over here. And that 6-carbon molecule is called citrate. Same as pyruvate was pyruvic acid, citrate is citric acid. And that is why it's called the citric acid cycle. So the other key things that we were talking about with this is that it is going to, we go from a 6-carbon molecule to a 5-carbon molecule, and carbon dioxide leaves. We don't need to keep track of the amount of times nor the names of all these. If you take a further, like a biochemistry course or another physiology course, you'll get into more detail, but let's just really focus on the framework of this. So we have the carbon dioxide coming out, and that's why we needed the oxygen for this stage. We also have these energy intermediates here. And so we have the NADH, and you can see the little electrons being carried by it, and the FADH2, and the little electrons being carried by it. And those are the intermediates. They are the things that are going to be used up in the electron transport chain that's part of oxidative phosphorylation. The other thing here is that we have a molecule of ATP. Now, it says in the earlier slide that we make two ATP molecules. And the reason that we make two ATP molecules, I'm going to make you dizzy, flipping all the way back to where we did that simplified drawing of glycolysis. When we ended off with glycolysis and then this transformation to acetyl-CoA, we had two molecules. So each molecule is going to enter the citric acid cycle, producing an ATP. So we get two different cycles going on, and we get a total of 2 ATP from the Krebs cycle, or from the citric acid cycle. And so that's really everything that we're trying to keep track of. We should be able to do a simplified version of this, kind of capturing everything that's on the words here, and really kind of stripping down that really complicated cycle into something that's a little bit more streamlined. So the key bits are that we're going to start with acetyl-CoA. And if you don't like writing the words, you can do a 2-carbon CoA molecule. If you wanted to, that's going to combine with a molecule to make citric acid, if that helps kind of ground you as to why it's called citric acid. And it's a cycle. It's literally going to cycle around in this process and come back to the beginning. So the key thing here is we've got oxygen being combined with any carbons that are coming off, creating carbon dioxide. So every carbon cleaving or removing step is going to produce some carbon dioxide. The other key thing is, so it requires the oxygen for that. The other key thing is that it's going to produce these energy intermediates, the NADH and the FADH2. And if those don't mean anything to you, remember their role. Remember that they are electron carriers or intermediate to the process. So energy intermediate. Intermediates. Okay, the other thing here is that it's going to produce an ATP. And the key thing that I said is that this whole cycle is actually duplicated. This is happening in the mitochondria. And we're going to take a look at the chart now to again simplify it in another way. So we've gone through a couple different ways. There's the words, there's the big scary picture or diagram, there's our simplified diagram, and then there's also this chart that really tries to drive home that this is the next cycle. So it'd be the Krebs cycle. Oops, I'm erasing instead of drawing. Krebs cycle. And I'll allow you to make the choice what you want to call it. Just be aware. So it's also called the citric acid cycle. Just be aware that sometimes it only has one of the names. And in this chart, again, you can remember the first thing was where does it happen? So it was the glycolysis was happening in the cytoplasm. We are now in the mitochondria. And the next kind of summary thing here in the chart, just trying to make it easier, is does it need oxygen? Like, yes, it needs oxygen. What goes into the Krebs cycle? So there's a bunch of things that go in, but the key thing that we summarized was this oxygen, right? Oxygen was required. And also the acetyl-CoA. You can get into the precursors that help you understand the formation of those electron carriers, or you could say technically ADP has to go in, but we're going to just keep it as simple as possible. Again, as a basic framework that you can then expand on in later courses. So what comes out of this stage? Well, we're going to have carbon dioxide come out of it. We're going to have the NADH or the FADH2. And you can think of them that way. You should also remember their role, that they are the electron carriers. Or the energy intermediates. And the real reason we're doing this cellular respiration is we're going to get 2 ATP. And again, it's one from each cycle because we have 2 cycles going on from one molecule of glucose. So the last stage, you've made it. This one is actually a 2-for-1 deal. So the third stage is oxidative phosphorylation, and that is a heck of a mouthful. I've always suggested that people name their potted plants or their kittens or whatever. Somehow roll this into your life so that you have at least tried to say that out loud. So oxidative phosphorylation. And we're going to talk about at the end how that name, it is actually descriptive, even though it's got a lot of letters and syllables in it. So it has two different parts. So the oxidative phosphorylation has the electron transport chain in it, and it also has chemiosmosis. So really these two stages rely on each other. The electron transport chain is going to set up chemiosmosis so that it's possible. And the chemiosmosis is the major payday. To this point, we've only made 4 molecules of ATP. Well, we're going to make, I don't know if you can see ahead, we're going to make 28 out of this last stage. So it's really important to get the details so that it makes sense. Again, we're staying with a summarized version here so that you can understand the framework of this, and then you can fill out more details in later courses. So during this stage, basically, we're going to use those electrons from those electron carriers. We're going to use those molecules that we called energy intermediates. The electrons from those are going to shuttle through this electron transport chain. And what the electron transport chain is a series of proteins that are found on the inner mitochondrial membrane. And you remember that the mitochondria had those 2 layers of membrane. And so where we're talking about this, where this stage is happening, is across this inner mitochondrial membrane. And so just for some context, we are now in the mitochondria. So across this phospholipid bilayer, there are proteins embedded, and the electrons basically get passed from protein to protein. So many words in this. And ultimately are going to fall to oxygen. The other thing that it does is it sets up a hydrogen ion concentration gradient. And we're a little scant on the details of how this happens, but really that it happens is where we're going for here. So the electrons being passed through somehow drives hydrogens to the outside of this mitochondrial membrane. The other thing that the electron transport chain does is that it's going to use oxygen. So this last stage requires oxygen. And we remember how greedy oxygen was for electrons. Well, we just had electrons being passed from protein to protein. Eventually, those electrons are going to be attracted to the oxygen. In the textbook, it says they fall to oxygen. And when we have oxygen and some spare hydrogens around, we're going to produce water. And water is one of the products in the overall chemical reaction of cellular respiration. And this is the only time that it shows up in the entire story. So it's kind of a key player. And it's easy to miss it, but it's absolutely important because it's one of those major players in cellular respiration. So we're going to move on from the electron transport chain because I said that it sets up this stuff for chemiosmosis, and specifically that hydrogen ion concentration gradient. So we had a different concentration of hydrogen ions on one side of the membrane than we did on the other. And anytime we have this concentration gradient, we know that things will move to equalize that. And so we can have passive transport of hydrogen ions. Well, those hydrogen ions happen to pass through an enzyme, happen to pass through another protein across that mitochondrial membrane. And that protein or that enzyme is called ATP synthase. Great name because what it does is synthesize ATP, which totally makes sense. And so this is that process of chemiosmosis. This is the process that is the major payout. This is the process that makes a whopping 28 molecules of ATP. And so, you're not really done the story of cellular respiration until you can make sense of this last stage, and the stage having, the electron transport chain that then sets up chemiosmosis. I've done all of these with a bunch of words, and I think it's important for different brains to think of this in a picture, and then we're going to summarize it again in that chart to try and get rid of so many words. So this is oxidative phosphorylation. If we take a step back on that big massive word that you're going to name the new potted plant in your house or something, it requires oxygen. And what it's doing is making a lot of ATP from ADP. It's phosphorylating. It's putting a phosphate group onto the molecule of ADP. And a lot of it, right? Like way more than the rest of the stages of cellular respiration. So that's why it's oxidative phosphorylation, because that's really descriptive to what's happening. It has the two parts. So it has the electron transport chain and chemiosmosis, and one basically sets up the other. So the electron transport chain starts with the electron carriers, the NADH and the FADH2, and this is that inner mitochondrial membrane. So it shows it here, the inner mitochondrial membrane. And so the electrons get passed through, and this is a bit of magic, I always said, with this yellow arrow, and I know we're not fully explaining how that happens. The electrons fall to oxygen and combine with more hydrogen that's present to make water. And again, this is a big deal here because water was one of the products of the overall cellular respiration chemical reaction. And this is the very only time that water shows up as part of this story. So water is important from the electron transport chain, but the other thing that's important is the setup for the chemiosmosis. So this is illustrating the setup. With the movement of those electrons through that membrane, it causes or forces hydrogen ions to be pushed to one side. So now we have a concentration gradient. We have more hydrogen on one side than we do on the other. Well, that's where we come into chemiosmosis. So that concentration gradient is primed to be equalized, and the hydrogen ions that pass through this other enzyme, this ATP synthase, are what drives that phosphorylation here of ADP into ATP. And like I said, this is the major payday. This is where we make 38 molecules of ATP. And this is why the story of energy production on a cellular level is not done until we can make sense of this final part of the third stage of cellular respiration. Okay, fewer words. Let's try and get it captured on my little chart summary here. And so in this one, the third stage is oxidative phosphorylation. So many letters. And it's going to have two parts. I'm going to make this one shorter because these are little simplified notes. The ETC and the electron transport chain and chemiosmosis. Both of these are going to happen in the mitochondria. And again, it's the inner mitochondrial membrane to be specific, but just to get it kind of streamlined and kind of a few keywords to kind of get your story flowing here. It's the mitochondria. Does it require oxygen? Yes, because it's oxidative phosphorylation. It requires the oxygen because that's key in the story of how the water gets made. So what we need going into this, we can actually divide these second two boxes into two parts, really kind of keeping track of the electron transport chain and how it sets up chemiosmosis. So what we need for the electron transport chain are electrons, and the electrons come from the electron carriers. This is also the key bit that requires oxygen. So you could call them electron carriers. You could be consistent, like I have been doing it all the different ways, NADH or FADH2. You could call them those energy intermediates. This is kind of where they were going. They were intermediate to the process, but now they're getting used. And the electron transport chain takes those electrons and it's going to do two things with them. So the first thing it's going to do is set up a hydrogen ion concentration gradient. And certainly you can make these notes useful for you. So I like the symbols, but you can write out hydrogen ion if you will. The other thing that it does is it's going to produce water. When those electrons fall to oxygen, because oxygen is just so greedy for electrons, and combines with some spare hydrogen ions, it's going to make water. So that's it really for the electron transport chain. A lot of magic in there, a lot of questions, definitely some rabbit holes to fall down. In class we can chase those down a little bit so it makes sense for you. But the next part, the chemiosmosis, is now going to use the hydrogen ion concentration gradient and ATP synthase to do this final little bit. And the final little bit is making all of the ATP, 38 molecules. And so this is, like I said, the major payout. Sorry, 28. Did I say 38? 28, because we get to a 34 total. Your textbook, just to give you a disclaimer if I say the wrong numbers, your textbook changed the numbers on here because this is still a process. They're still trying to figure out, you know, what is the average efficiency and how many ATP molecules are being generated from one molecule of glucose. And so, yeah, I think, I can't remember now the last one, but so I see, the last one was 34. My goodness, I'm confusing you. I think this isn't a math class. So 32 is our new total. And so, you know, the big idea here is that this is a whack of ATP that gets produced. Okay, so we're going to head back and finish this off. In class, I'm sure people can heckle me. So this is just illustrating, if it didn't make sense to you, the two parts of the electron transport chain. So the electron transport chain does two things: sets up the hydrogen ion gradient and produces the water. So as a summary, as an overall kind of how it all comes together, we have the three stages. We have glycolysis, we have the citric acid cycle, and we have oxidative phosphorylation. That is both the electron transport chain and chemiosmosis. Now, they happen in different places. So the glycolysis happens in the cytoplasm, where the other two happen in the mitochondria, right? The key things that come out of glycolysis is that the glucose goes in and the pyruvate or the pyruvic acid comes out. We are producing two net molecules of ATP and some of those energy intermediates. We've got some NADH that's carrying electrons for a later stage in cellular respiration. This little arrow here is talking about that in-between stage where we went from pyruvic acid or pyruvate into the acetyl-CoA, using some oxygen, not technically part of glycolysis, and producing our first molecule of carbon dioxide. The acetyl-CoA is going to go into the citric acid cycle. The citric acid cycle is going to produce ATP, 1 per cycle, or a total of 2 for 1 molecule of glucose. Quite a bit of carbon dioxide, and again, these energy intermediates or these electron carriers. Into the last stage of cellular respiration, these electron carriers are going to give their electrons to the electron transport chain. The electron transport chain is going to set up a hydrogen ion concentration gradient for chemiosmosis, and those electrons are going to ultimately end up with oxygen and form with hydrogen ions to make water. Chemiosmosis picks up with that hydrogen ion concentration gradient and ATP synthase to make this big payout of ATP. I'm going to get the numbers right here, 28 molecules of ATP. And there was the water. So if we kind of take a look at these, there is a divide. The first stage, the glycolysis, can happen without oxygen. So that technically is termed anaerobic, no oxygen. Aerobic is requiring oxygen. And so these later two stages, as we filled out in our chart, do use the oxygen. This is a summary. I think I just talked through most of it. The key thing here is that it is about 40% of the glucose potential energy that's coming out. The rest is put off as heat, as we know. And really what we're trying to do here, you'll notice that this page does not have any of the intermediates. It only has the reactants and the products of respiration. So as important as those electron carriers are, they aren't on this page because they're intermediate to the process. So they're necessary for making sense of the story, but they're not part of that chemical reaction of cellular respiration. So when we understand cellular respiration better, then we can understand how certain poisons or drugs can actually work. So there's three different categories of poisons or drugs that work. So the first category is going to block the electron transport chain. And if you block the electron transport chain, then you're not setting up the hydrogen ion concentration gradient. You're not setting up that big payout of the chemiosmosis. And so insecticides, cyanide, and carbon monoxide are all examples of toxins that work on that level, on that cellular level. The second one is going to inhibit that protein, that enzyme of ATP synthase. And because that was the big payout of making the 28 ATPs, well, those cells are going to be deprived of the energy in the ATP. And so the example here, not a toxin or drug for you, but or toxin or poison for you, but actually a drug for you. So if you are a bacteria, then oligomycin, an antibiotic, works in this way and would cause damage to the cells because the ATP synthase isn't working. So the prokaryotic bacterial cells that are making you sick are not going to be able to live because they're not going to get enough energy for their own needs. Finally, the third one makes that inner mitochondrial membrane leaky, so it can't get pumped up with that hydrogen ion concentration gradient. And this one, it's in the textbook, a diet pill in the 30s. Like, yeah, that would make you lose weight, but it would also make you die a little bit. The other one here is wood preservatives. So, you know, they have changed pressure-treated wood and tried to make it safer for reasons like this. You don't want your kids playing on playground equipment that's got toxins in it that make their cellular respiration not work. And so this is just illustrating those different places. So either the electron transport chain for the rotenone, the cyanide, and the carbon monoxide, or the ATP synthase for that antibiotic, the oligomycin, or the membrane being permeable or leaky to the hydrogen ion for that the diet pill that was noted in your book. So what happens when we don't have oxygen? And so what happens when we don't have oxygen is lactic acid fermentation. So we talked about how regularly we're talking about cellular respiration, and it is an aerobic process, but we have options. So if your cells are short on energy, so the need for ATP is greater than the amount of oxygen that can be delivered, then muscle cells are going to switch into lactic acid fermentation. It's a way of still getting some ATP to keep the cells working. Outside of humans, it is in cheese or yogurt, and it is that kind of tangy taste because it's acidic. And really what's happening, I like the picture of this one. So we know that glycolysis doesn't require oxygen. And we also know that there is a slight ATP payout. And so what lactic acid fermentation does is just recharge what's required for glycolysis, this precursor to the energy intermediate, so that we can keep pumping out some ATP. And the byproduct of that is lactate or lactic acid. And it's what you feel in your muscles when you run up the stairs because you're late for class or run for the bus or whatever it is. Contrary to or different than this would be fermentation, alcoholic fermentation. So again, not in humans. Your notes are not in color. So only the digital resources are in color. Again, this is not in humans, otherwise running for the bus or up the stairs or going to the gym would have other adverse side effects. You would be drunk. And so this is, again, not in humans, but it is in yeast and bacteria and some fungi that we use in winemaking and beer brewing, because the byproduct, the same thing happens, like it can still derive ATP with the glycolysis, but the byproduct is different. Instead of lactic acid this time, it's alcohol, it's ethanol. And so that's the process. It keeps the cells alive because you get some amount of ATP production, but the byproduct is alcohol. The last two slides are really just to say we focus so much on glucose and its path through. To pick up on where we left off with the macromolecules in chapter 3, there's a whole lot more going on. So all of the macromolecules can actually have a place to play through this process. And really that's what this slide is trying to illustrate here. The other thing is that, that interplay. So we're not just breaking things down for energy, but we're using that energy to build things back up. So it's the breakdown and the buildup that is required to keep your cells alive. So again, just kind of zooming back out a little bit to see the context of why you're producing ATP. It's so that your body can then use that energy to do all the things that it needs to do. And synthesizing molecules is also part of that. So these last slides are just really meant to illustrate the interconnection of a lot of the things that we've talked about so far and how the cells do what they do to stay alive.